Can everybody hear me? I'm going to take the fact that there's a recording progress to mean that you can hear me. So welcome everyone to our KCART Autism Research Seminar this week with John Colombo. I'm going to briefly introduce John, although I think probably everybody here knows John or knows of John. So, but for those of you who don't, he's our interim dean of the College of Liberal Arts and Sciences, emphasis on the interim, right, John?
Right.
And he is the director of the Lifespan Institute and professor in psychology, active member of the Brain Behavioral Quantitative Science program as well. It's exciting, and I appreciate John being with us today. As many of you know, John has done some seminal work in infant cognitive and attention development, including development and initiation of multiple clinical trials looking at different nutrients and micronutrients for that are related to infant brain development. Some of these studies actually have been key work supporting integration of DHA in baby formula, which really, I think he may talk about some of that today, but yeah, it's just a beautiful example of how basic mechanistic science can actually be translated to change public health and policy as well. And it's a great model, great example of that, and a goal that many of us as researchers aspire to emulate. So it's going to be great to hear about that. Real briefly, I have to brag about John's accomplishments. He has over 120 peer-reviewed articles published, 6 books and monographs. He's been cited 12,000 times. It's a big number. He's also accrued greater than $30 million in extramural funding across NIH, NSF, private foundations, as well as industry. I have to say he's also responsible for many of us being here at KU. He's been here since he was a postdoc. And I'm not going to tell you
years.
200. That's
200 years.
I wasn't going to say the year anyway, so don't worry about it. But yes, he is a pillar at KU, and many of us owe quite a bit to him. So I appreciate all of that, and I appreciate him giving this talk today. So today he's going to talk to us about adventures in fish oil. And so if that doesn't hook your interest, I don't know what will. So thank you again, John, and welcome.
Thank you, Matt. That was it was way too kind. And there's a whole lot of other things Matt could say. I wanted to thank him for that. And it's really nice to be here. Like, spent the last 6 years chained to my desk as an administrator, first at Youngberg and then at Strong Hall. And I still chafe when people introduce me as an administrator because I still identify as a scientist. And it's nice to be able to at least get up and talk about what the science that we've been doing for the last 25 years or so. So yes, we will get back into the last century. I really want to emphasize that this is that the emphasis here is that on sort of how you can start out as a basic scientist and then have it sort of have that impact compounded by simply being willing to move into applied settings. And so that's pretty much it. See if I can get this to go.
Click the blue got it button.
What button?
Do you see the? Do you have that message?
I do not have that on there.
Oh.
So I don't know who's got that. Is that you? Let me see if I can.
I think, John, having done this earlier, you need to move your mouse over to that screen and click on it. Like the mouse goes across.
Does it go across?
Yeah. There you go.
I have to aim right.
Almost.
This is like,
Yeah.
is that it?
Yeah. There you go. Now you should be able to.
Okay, great. There's like those movies when you have the person who can't fly a plane in the cockpit. So a lot of people don't know, since I've been in administration for a long time, people don't know what I've been up to, and I just kind of like to encapsulate what research is about. Continuing to with postdoc to work with Francis Horowitz. We really want to be president of the community graduate system. And it was really sort of formative in pushing to do work on individual differences. And she was all about identifying kids at risk, and it sort of fit really well with what Dick Schieferbusch was doing with respect to intellectual and developmental disabilities. I really got into the concept of measuring individual differences in babies. And what I was looking at was neurocognition. Things that were not cognition back then. It was perception and sensation. But what did it mean? And that was Francis' question all the time. Well, this is great. What does it mean? What does it mean for the long term? And so I really tried to follow her out and follow that emphasis and really work on refining the rules of neurocognition at early infancy.
Hey, John.
Yeah.
John. Hey, I'm sorry to interrupt. Can you actually move closer to the podium? We played with the video, but the audio is going in and out for those of us who are on Zoom.
I think it's. I think that's it.
Okay. So is that better for everybody?
It's actually not that much better.
Is it not much better?
That's a little better. That's a little better.
So did was early on in the last century, it had to do with looking at psychometrics of these early neurocognition, because the idea was that if they weren't stable or reliable measures of individuals, then they really weren't worth anything. And so what we're trying to do here is see what of these psychometrics could be used for early identification of risk or all sorts of developmental problems. And the last thing, were they predictive? Were they meaningful outcomes? And what were the mechanisms underlying them? So that's what I was up to. That can you hear me now? Is it better? Can you hear me? Can't hear me? Okay, let's see.
No, we can't. Cassie, can you maybe, oh, you know how to change the mic? If not, we might need to get Brian back in. Yeah, that'd be great. Thanks. I'm just trying to send you a text.
Hybrid models. Here's let me locate my mouse. Sorry. Hybrid is hard. How's that? Is that any different?
Is that any better?
Better?
Much better. Much better.
Oh.
Can't receive or sound now.
Sorry. The first one.
Kate's got it.
Okay, so, okay, yeah, you need to change the speaker then, so you can hear them, try that one.
How about now?
You guys try talking?
That sounds good.
No. Okay, there's a
Sorry, it's
couple
just hard for me
different
because I can't see.
It's only really two speakers. Yeah, I was gonna say, if they can hear John, maybe that's.
How about now?
Can you guys hear us, but can you, what have you tried talking?
We can hear you and we can see the slides as well.
You guys.
Maggie's muted, but nobody else.
But Matt's not. and
Matt's
he's
not.
trying to-
It's so strange.
So it's speakerphone.
I tried to change it. I'll change it back again.
Do you have anything like the sound muted on this computer or on the TV?
No. can try to grab Brian.
Oh,
Sorry.
you can change it from say assistant. Go down to audio, so it's gonna go all the way down. Your podcast.
Yeah. So it looks like the mic is going, but speakers.
Yeah, I think they record the one.
Test.
Yeah, yeah,
Right, these are all recorded.
yeah. Here,
Yeah, try that. Should make noise if it's doing it right. So try flipping to the other one.
Can you hear us?
Because we should hear like a.
got it.
Sounds like a series of
Hi,
Ryan's a rescue.
yeah. I think they can hear us better, but we can't hear them.
I'm not sure.
We tried it with speakers.
Yeah.
Zoom controls are all I know.
Should be better. All right.
Yes. Matt, can you try talking now?
Yeah, can you hear us?
Yeah, very loud and clear.
Can you hear us?
We can hear you. This is great, amazing. All right. Thanks,
All right.
Brian. Thanks, Cassie.
All right, thanks, Brian. There's beer in this for you. All right, so I left off, so this is what we're doing. The idea was to really sort of hone in on individual differences in these measures of early neurocognition. Are they predictive? Are they reliable? And are they really, what's the mechanisms underneath that?
Sorry, one more time and flip the slide.
Let's try that. All right, yes. Back, I think. Nope. All right. So do I
Just click somewhere.
there?
Now try.
There you go. All right, yay. All right, and then once you know how to do that, you're really then into the place where if you have a measure that's meaningful, you have a measure that's predictive, people come after you because they want to run longitudinal studies on their interventions and they want to know the answers before they die. And so they want to get a measure during the early development that will tell them whether their intervention's effective. So people came after me in sort of the late 1980s, as far back as that, asking me to be worked into their longitudinal studies. So that's pretty much it. Okay, hopefully we'll move from here on. I really started out working on infant visual habituation, which is a really nasty little paradigm where you take babies, you tie them up and you show them pictures until they can't stand it anymore. And we augmented looking time with heart rate, and this is a baby being hooked up with EEG, ECG electrodes. You basically put the baby in a car seat, you turn them around, and then you have them look at a stimulus. And while they're looking at the stimulus, you are looking at them, and you're coding their eye movements. And while you're coding their eye movements, you're also getting a measure of their heart rate while they're looking. And you'll see a little bit that when babies do pay attention, when attention kicks in, there is a really robust parasympathetic response that also gives us a notion of the quality of attention. So that's what that's about. If you show a baby a stimulus over and over and over again, much as if you present any stimulus to any organism at any level of phyla or development, you do get a decline in the response. And the decline here is in looking time. As you can see, these are curves for babies from 3 to 9 months of age. This is really early work that we did in the late 1980s. looking at the developmental course of looking, and as you can see, the major thing is everybody drops, they're looking, but what happens is that the major developmental thing that's happening here is that their looking time from age to age tends to drop. So 3-month-olds look for a really long time, 4-month-olds look for less, 5-month-olds less than that. And by the time you're 7, 8, or 9 months, you're pretty much not looking for very much in terms of looking time durations. If you plot this the other way, this is a plot of looking time by age. Age is along the x-axis here. And this is a compilation of any number of studies that were available in the early 2000s. You can see everybody gets this. Everybody shows that looking time drops with age. So if looking time is dropping with age and you're getting better at something with age, it makes sense that you're actually processing information more quickly. And that's really where we came down on things. And there are other things that affect looking time or that are related to looking time. But for the most part, it's all about how quickly babies process information. So the developmental course is to drop. The lower you are, the faster you are. And so that's basically the idea there. Now, if a baby looks for a long period of time, is that something that they do across ages, or is it reliable? And we published really one of the landmark studies in psychometrics of this basic learning paradigm back in the 1980s. And here, these are simple zero-order correlations within age, and you can see that starting around 4 months of age, the duration of looking is pretty reliable overall. It's not super great. It's not like an IQ test or anything like that, but they are relatively stable. And if you look at on the longitudinal study, the stability over time, once again, it's not super high, but there is a fair amount of reliability there, a fair amount of stability. And in fact, they are getting you are getting some notion that babies who look for a long time at 3 months end up looking for a long time on average at 7 and 9 months of age. So this turned out to be really interesting. And the other thing about it was that it was actually predictive, as you'll see later on. Now, this is the parasympathetic response that you get when babies are actually looking. This is a stylized plot of looking time, which is within the arrows, the blue arrows. And then you have heart rate here on the y-axis, and you can see that there is a little bit of a blip when the baby starts to look, a little bit of heart rate increase, and then there's a prolonged period of deceleration. That deceleration starts to end, and you start to get a return to baseline, and then the baby stops looking. It's really actually quite predictive. If you're standing there and you're watching the look occur, and you're watching the heart rate, the EKG, what you'll see, you can turn to the mom and say, your baby's going to stop looking here in a minute. And sure enough, that always happens, because the return to baseline actually predates predates or presages the behavioral response. And you can look at that in different ways. We look at the amount of sustained attention that occurs during that decelerated period, as well as orienting, which is the first part, and attention termination, which is the last part there. What's predictive here is on the behavioral side and from the heart rate defined attention side are these things. If you have shorter look durations and you have a robust age-related decline in looking, that is an indication of sort of typicality or normality, if you will. The other thing that happens is that in babies who score really high later on standardized tests, their sustained attention remains at a fairly high level across ages. Their orienting sort of goes up and their attention termination drops. And attention termination is when the baby's continuing to look, but their heart rate is returned to normal stages. It's sort of like they're looking but not processing. So that goes away. As you might expect, if you're looking at less optimal outcomes, what you have are longer look durations and really not such a great big decline developmentally. And then you'll find often that in these kids that sustained attention declines across age, and you get increases in both orienting and attention termination. That is that they tend to be slower
to decelerate, and then they tend to keep looking even after the deceleration's over. So these are the things that we have that are actually really pretty predictive. Once you have this, once you have a measure like this, people come out of the woodwork and they want to talk to you about including your work in. And so I started to, as early as the late 1980s, I was contacted by people who were working in the area of iron deficiency, and they want to look at that with respect to this measure. But I started to work with Sue Carlson when Sue Carlson was Susan Carlson, who is now at KU Med. She was at the University of Memphis, at Tennessee at Memphis Medical School. And she was working on something called docosahexaenoic acid. And this is one of a number of long-chain polyunsaturated fatty acids. And I had to do a lot of reading when I started to get pulled into this literature. But there are two major LC-PUFAs, long-chain polyunsaturated fatty acids. One is arachidonic acid, and the other one is docosahexaenoic acid. So it's ARA and DHA. And ARA is actually, they're both good for you. And but in different ways. ARA helps with sort of neural repair and sort of sustenance and growth. DHA actually is part of the neural receptors and has to do with processing catecholamines. And the important thing about this is that there's plenty of ARA. We get it in all our diets. If you eat bread or meat or in leafy vegetables, it's all in there, or grains. DHA actually, we don't make it, and you have to get it predominantly from your diet. And the way you get it is from the oil that's in fish, and that's why this is a talk about fish oil. So, and there's plenty of ARA in the diet, but DHA you have to get, and babies can't make it, and so they get it from their moms, or they get it from their diet. And we've known for a long time that if you give DHA, you actually have improved kinds of sensory functions along the way. DHA accumulates in the brain very early and at a very high rate. And if you, and this is just a way to show that it's an integral part of the central nervous system. It's in the retina, it's in the brain, and it accumulates at a very high rate. The red line down there shows prenatal accumulation. And then it sort of levels off as time goes over on, but it's still getting in there. And we were especially interested in this early accumulation because you had to have it. And so it looked pretty interesting because babies have to get it through their diet. They accumulate it it goes right to the brain. It contributes to brain development. But on top of that, American babies don't get very much of it because we don't eat a lot of fish, and we don't eat a lot of the things that have it. And so it's interesting. For example, U.S. mothers, their breast milk is actually quite low in DHA, whereas mothers from around the Pacific Rim, sort of northern Canada, all the way around to Asia, actually have it really pretty high. We knew for a long time that if you measure infant visual acuity, now DHA is actually a structural component of the photoreceptors in the eye. If you feed DHA to preterm infants, you actually get improved vision, visual development, visual acuity. This has been known probably since the early 1980s. And what's really interesting here, and this is just a plot from a study, and here a lower score on the y-axis means better acuity. And the top group here, the top line there represents an unsupplemented group, and then the three lines below represent groups of babies that were supplemented with different levels of LC-PUFAs in their formula. It improves vision, but the problem is that for pediatricians is that as they looked at it, if you start to follow these kids out by 2 or 2.5 years of age, they all pretty much end up the same. So for pediatricians to look at this, and this has been an education for me working in the translational field, pediatricians, if there's an early advantage, pediatricians don't really count that as being very important if everybody catches up. Whereas as a developmental scientist, I know that if you're ahead in something, it lets you do other things better, and then it compounds over time. And trying to teach that to healthcare professionals is not the easiest thing to do. So I spent a lot of my time doing that as well. Now, what was really interesting is this. In the late 1990s, when people started to come to me and say, hey, we've got this looking time thing. What does it mean? There were two seminal studies that basically used our measure that was developed here at KU. One, and it was Susan Carlson when she was still at the University of Tennessee. And then there was a group at Oregon Health Sciences University in Portland. And Susan was putting LC PUFAs into preterm formula. And the group and this is Martha Nuringer at Oregon Health Sciences University. They were taking DHA out of, or LC PUFAs out of monkey diets. And what happened is that if you supplement the preterms, they look for shorter periods. And if you take the substance out of the monkey diets, they look for a long time. And so when these things were published, I don't know if you can see that, but I've just highlighted that infants who were supplemented looked for shorter. That means they were more mature, and monkeys who were deprived of it looked for really longer periods. And so that implied that not only was DHA affecting visual development and visual acuity, but it was also maybe affecting cognition. And once this hit and people started to think about this, we got a big change in the formula makers. So both Mead Johnson and Abbott Laboratories pretty much announced on the same day in 2001 that they had decided to add lipids to infant formula. Now, lipids had been in infant formula in Europe for a long time because it's part of the complex in breast milk. U.S. manufacturers were not keen on putting it in there because it affects the shelf life of the product, it increases the price of the product, and so there were all these complications it produced. But I think it's an example of how if you do basic research and it gets caught up into the more translational part of the complex of science, you can really have an impact. And so ever since then, I've been working with food companies on the side and infant nutrition companies on the side, and really working with them to help them evaluate the different changes that they've made to formula over time. But it's kind of rewarding when I go down the baby aisle and you know, like you know how old I am. We don't have any more kids, but it's nice. I take a detour down the baby aisle and I just look up there and it's like, yeah, we did that. So they and of course there was a little bit of a backlash from folks who were like, oh, you're trying to make baby formula like breast milk, and that's not the case. In fact, breast milk is most moms actually end up formula feeding even if they want to breastfeed because of the way that our modern society. And I've got a whole spiel on that, but we're a little late getting through this, so I'm just going to leave it where it is. But this was a pretty widely noted around the country as one of the things that we've done. Then we started on, well, really, how much do you need? Is it, and when do you need it? The first study I worked with Susan on in 2004, in the early 2000s, really was a follow-up of studies where she fed high DHA eggs to mothers who were pregnant. And what we found there, quite simply, was this: babies from moms who had high DHA out of that sample looked for shorter periods and declined really pretty abruptly, while babies who were from moms with low DHA actually had longer looking. And when we followed them up at 18 months, we also found that they were 12 and 18 months. We also found that they were actually much more distractible on an attention span task. So it looked like DHA, prenatal DHA, early on, you know, that prenatal DHA levels in the mother were affecting the baby's outcome. And so this really got my interest up because I had been drawn into this literature kicking and screaming, not really believing this was going to work. And suddenly all my interests in critical periods and sensitive periods and fetal programming and all of that started to come to the fore. So that launched our laboratory, and this is our lab at the KU Med Center. Susan's in the dark dress to the right, back right. Kathleen Gustafson's in the green, and Byron Gajewski is our statistician, is leaning up against to the right there. This was our group. At various points in time, we had a group that was as large as 18 or 20 people doing research on these things, and all those things listed at the bottom are clinical trials that we've run, either NIH supported or supported by industry over time. We were lucky to have, and I'm not going to talk about all of them, but I'm just going to talk about the neurocognitive pieces in some of the studies that we've done. First one I'll talk about is the Diamond trial. We ran this for a long time. Mead Johnson funded us to do this. And we used, the question was how much DHA, how much LC-PUFA do you need in formula? Because the idea is that if you could put less in and still get the same effects, it would actually be a benefit to know that, because you don't want to overdo something. And also it has business implications as well. And we fed babies an unsupplemented formula, which, by the way, can't do anymore in any research. It's not ethical, which is also a measure of your impact, right? We fed them an unsupplemented formula for 12 months, and then we fed 3 other groups different levels of DHA and ARA for 12 months. And we followed them through 18 months, and then we extended the study after that twice to look at ERPs and then outcomes later on in neuroimaging. What we found was that babies who were fed LC-PUFA supplemented formulas, and I'm simplifying here so everybody can just kind of follow it. This is the regression line for sustained attention from the attention task. And as you see, the orange line, which is the supplemented babies, basically were high and flat. Just as I said, that's the good outcomes, right? And babies who were not supplemented actually declined in their sustained attention. And so this is again, so it's really an experiment where we change the quality of the attention that babies have, which was kind of an important and interesting thing. And so that's the sustained attention over the first year, whereas here it dropped off. And then when babies got older, we started to administer a bunch of these executive function tasks. And 2 that I'll point out is the modified Stroop tasks, which mean that you have to actually, you're presented with a task, and then you have to, which would typically elicit a normal sort of automatic response, and then you have to inhibit that automatic response and give the other response. And then there's this dimensional change card sort, which is built on the Wisconsin General Test Apparatus and discrimination learning tests that some people might be familiar with, but I'll show you what they are here. The Stroop here is the Stroop tasks. You show baby, or these are kids who are 3 to 5 years of age. You show them a card and it's red, or you show them a card and it's yellow. If they see red, they have to say banana. And if you show them a yellow card, they have to say apple. So the idea is that, and you play it up like, we're going to play a game, and I want you to fool me when I show you the red card, which would normally be apple, right? You'd say banana, and the other one, and if I show you a yellow card, you have to say apple. And so they have to switch that. The other one is there's a picture of a sun, and they have to say night, and there's a picture of a moon, and they have to say day. So there's the day-night task and the red-yellow task, highly originally named. Across both of these tasks, the supplemented kids did better. And you can see that there, it's a main effect, it's highly significant. And that came out. And the dimensional change card sort, you give these kids, I keep saying babies, you give kids a card that can be sorted on either color or shape. And you reward them or you give them good feedback if they sort correctly, and you tell them they're wrong if they sort improperly. So you pick one and its shape, and they sort on shape. You say, that's great. And then they learn pretty quickly that you want to sort on shape, or you learn pretty quickly to sort on color. And then what you do is after they reach mastery on that, you then switch the dimension. Say, okay, now we're going to do the opposite thing. So it measures their ability to learn the rule, their ability to inhibit that rule after they've learned it, and some sort of mental flexibility. And once again, kids the control group here didn't get better, that's the dotted line, didn't get better at this task until about 5 years of age. Whereas we improved the supplemented kids, they got better at it. They're about a year ahead on this task. And so this was really, really, really interesting to us. When we followed these kids up on IQ scores, these kids were coming from a very low SES population. We essentially normalized their IQs at age 5 and age 6. There were big effects on IQ, and Matt knows this and Carrie knows this, but if you've ever tried to do interventions to change IQ, it's not easy. There have been people who have died trying, and I was amazed to see this, honestly. Everyone asks why is the control group at 85? Parents' IQs from the sample is about 88. And so that gives you a sense of where, and I put the effect sizes up here just so you can see what those D primes are. We then follow these kids over again. We published a paper in Developmental Science showing that ERPs were different. We got a lot more frontal function and we on ERPs and higher amounts of synchrony. And this neural synchrony is the thing that sort of ties the brain together and produces semantic networks as kids get older. And then when we did some work at Hoagland, I see Bill's on the wire here. It's good to see him again. And we found greater white matter, which is myelin, for kids who were supplemented as well, and lots more connectivity, which is the thing that underlies executive function. To be honest, it's an integrative function that develops over time because the brain gets wired back together and self-organizes. So that's the diamond study. One of a number of studies that we've done, but basically showing that if you feed kids a supplemented formula from birth to 12 months, you get effects all the way out to ages 6, 7, 8, and 9. And again, the idea that this early period is really important, the first 1,000 days, right? Just because we have this, you can see this, the ERP studies. Here, these are microstate analyses for, we did a go, no-go task where you have to inhibit, and inhibiting involves the frontal cortex. The control group over time, basically when they're involved in an inhibition trial, you can see that's where things light up and they go back to normal. The supplemented groups show far more frontal activity before we're running back to normal here. So again, evidence that the formula that we fed them really changed the structure and function of the brain. And what's of interest to us is that if you read the literature on LC-PUFAs, there's numerous meta-analyses that suggest that there's no effect. Those meta-analyses are predominantly based on the Bayley Scales of Infant Development, which is a global test of developmental status. Can your baby sit up? Can they crawl? Can they put blocks together? Things like that. While we were getting these sophisticated outcomes with laboratory tasks and granular and neurocognitive outcomes, the Bayley continued to be insensitive to these effects. And we've seen this in study after study after study. The global developmental tests typically are not always sensitive to these kinds of effects that we see, even when we're getting huge effects on these laboratory tasks. And it's just if you run into people who say, well, that doesn't really matter. Well, this is the answer for why that is. I've also been involved in a bunch of other trials and that are coming to fruition. A lot of them are showing positive effects. Some of them we haven't, not that as extensive as the diamond trial, but I'll just share with you some of the results that we've done. I've done with Me Johnson in Shanghai. There's the nice thing, if you go do clinical trials in China, you can get thousands of kids really quickly. And it's a really interesting model for how they do it. And we work with universities and hospitals in Shanghai to get this done. And we're mostly interested now in other things besides LC-PUFAs. And I've worked with zinc, I've worked with copper, I've worked with choline, lots of other different kinds of nutrients. But people are now interested in milk fat globule membrane. And all my years in graduate school, I never thought that I'd be even saying that. But in normal milk, which is the top panel there, the globules of fat are actually surrounded by a fairly rigid structure of nutrients that hold the milk globule together. And if you homogenize milk, or if you just take the fat out, you end up taking out that membrane. The membrane has all this good stuff in it. It has sugars, which are important for prebiotic function. It has cholesterol, which actually is important in the development of myelin. And it also has a fair amount of lipids in the membrane as well. Companies are interested in putting these milk fat globule membranes, or MFGMs, back into formula because they're normally in breast milk. And so we randomized, and this is the other thing, these are all clinical trials. These are double blind. Nobody knows who's in what group. You get the data, you run the analyses, you don't know what group is what, you draw your conclusions, and hopefully the results are coming out as you expect. We took babies and we randomized, and this is, I think, about 1,400 kids, randomized to a cow's milk-based infant formula or a similar infant formula with both MFGM and some amount of iron, lactoferrin in the formula. And these results are just coming out, the neurodevelopmental outcomes. We did use the Bailey here at 12 months, and it's the newer Bailey. We did use the MacArthur, and then we did it with Whipsy. And we actually, on the Bailey, we actually did see effects at 12 months on cognitive, language, and motor subscales. The Bailey no longer, by the way, for those of you who might know, the old Bailey used to have a composite, you had to measure, but now it's only presented in terms of these subscales. And by 18 months, it went away. But what's really interesting, I don't have it right now, but we got the Bailey results at 12 months. They went away by 18 months. So once again, the 18-month data will probably show up in some analysis if this doesn't matter. But at 18 months, we got really big effects on the language. And what's really neat, and so this came out just 2 years ago. It's under review now, but we followed these kids out to about age 6 or 5.5, and their IQs are higher. Who've been fed this MFGM. This is one of now 5 studies in the literature that suggests that MFGM actually has pretty powerful effects on neurocognition. And once again, we only fed these kids for 12 months, and we're seeing these effects at almost age 6. So it's a really powerful intervention. And it also speaks to the evolutionary intelligence of breast milk, because this is how the nutrients are constructed. So that's pretty much it. I tried to leave some time for questions and comments. You walk out of this, I hope you remember one thing, a couple things. One is that we can do valid assessments of neurodevelopmental outcomes using both standardized tests if you want to, but also, measurement is the thing. If you're a behavioral scientist, understanding measurement, you have a lot to offer to a lot of audiences in terms of if you know your measures, and measurement has been the thing that I've been called on time and again to help with in consulting with food companies or consulting with industry. It's just something that we really know a lot about in psychology, and we don't always sell it to our students, and we don't always sell it to our junior colleagues. the measures have their advantages and disadvantages. For example, you have to educate pediatricians on the neurocognitive measures that you use in the laboratory because they have no idea what that is. They have no idea what executive function is. You have to explain it. So the messaging becomes important. And so here's where science communication is rounding out to be a really important part of what we do in the next century. And lastly, I hope I made the case, and I've got lots of other examples. I couldn't fit them all in 45 minutes, but where if you do prenatal interventions or early postnatal interventions, they have really powerful and long-lasting effects. We don't know the parameters. If there is a sensitive or critical period for this, we don't really know the parameters of it, and it's not likely that we're going to be able to define them. Furthermore, when you work with nutrients, there's all sorts of problems that you have. It's really, if you're interested in mechanisms with human subjects, understanding why does this work the way it is, it'll drive you crazy because there's no way to do the fine-grained granular studies to see what the mechanisms are to follow. So that's where, for example, the preclinical work, the animal work is really, really important. The last thing is that when you work with, you get lots of different results. You get mixed results. If you do research, for example, on statins, you're working with a population who needs statins, right? You don't just go out and you randomly give statins to the world, and then you see whether or not it really affects the incidence of heart disease. You take a group that has heart disease, you give them that. In a lot of the nutrition work, we are doing the latter rather than the former. The former rather than the latter. We are going out and we don't know really the status, nutritional status of folks, but we're giving them these broad-based nutrients, and we're hoping that it makes a difference. And when you sometimes get a difference and you sometimes don't, you have to evaluate that in terms of whether or not you're really trying to improve the nutrition of a kid who's already getting good nutrition. And so that's one of the considerations. So those are the things, those are the lessons that I learned doing this over the last few years. And I really appreciate being asked to do this. And if you've got questions, I need to make sure that I give credit where it's due. All this work is done under a team and a collaborative team that has been really wonderful to work with. Everybody has their roles, everybody has their lanes, and we all respect each other. And it's been just an absolute pleasure to work with this group over the last 20 years. So thanks a lot. And I'm here for questions if you want.
Thanks so much, John. I can't see the hands in the room, so I'm not going to be able to call on people there. So I'll just ask you to do that, John, as a speaker, but I unmuted first. So I'm going to ask the first question. So, but I have a sort of a broader question. I think it would be helpful for myself, but I think there's probably many people curious about this. You've had this balance of funding larger clinical trials through both federal agencies and the private sector and industry.
Right.
And if you could just talk about how that gets balanced, right? Because the studies that you do through NIH are very different than the studies that like industry wants to do. And frankly, one of the things that's really impressive and I guess a bit inspiring too is this emphasis on measurement, because so many of the clinical trials that we see for those of us who study neurodevelopmental disorders, like you have, they use these really crude measures. And we've seen it within the autism world, especially, and different rare genetic conditions. These trials fail because they use gross cognitive IQ measures. But you've been recruited by industry because of the sensitivity and because you develop these quantitative approaches. So maybe if you could talk about that process, but also how you balance NIH and industry work and gone back and forth.
So NIH is pretty straightforward. Like any other federal sponsor, you submit your stuff, you hope for a good score, and you get it, or not, and then you do it or not, and you propose it.
It feels a lot more complicated than that, I'll be honest, but yes, I hear what you're saying.
It's a 2x2, but rarely do you get the grant not do it. So that cell is just forget it. Industry is an interesting thing. If you notice, a lot of the a lot of them, the things that were funded by industry. Is they're not interested in mechanism. They're not interested in what's the effect of this nutrient? So for example, when we did the Lighthouse study, it's MFGM plus lactoferrin. Why? Because Mead Johnson has a formula that has those two things in it. So the scientist in me wants to do, I want to do MFGM, lactoferrin, both, and a control. Well, that would mean 4,000 kids, given the power that they want to have, and we can't do that. So they're often, so you have to kind of put the science, the real, and you have to come to some and make peace with the idea that I'm not going to know maybe what the active ingredient is. You sometimes can figure it out. For example, in the Diamond trial, we think we know it's DHA because we can see the effect of DHA separate from ARA because of the way the doses came out. But that was just luck. That was just luck being able to do that. So you have to know what, in order to work with industry, you have to know what the industry, you have to sort of understand why industry is doing it, why industrial sponsors are doing it. There are some, and I will say this, there are some industrial sponsors I'll work with and some I won't, because there's often a negotiation about, well, we've got you to do this. How about if you do this too, and this too, and this too? And they start to load up on their clinical trials that way. And that, and suddenly you're doing a bazillion measures and you just put this stop and say, stop, I'm not doing this now. That we want to play by the rules. Here's the outcome we want. And sometimes they'll listen to that, and sometimes they won't. So different companies are really different, and the presence of a global medical officer is often a good sign that they're really interested in the research, truly the R&D piece of it, as opposed to just sort of, or where your money is coming from, whether it's coming from the R&D part of the business or from marketing. That's the kind of things you really want to pay attention to. The last thing is sort of who owns the data and what are the constraints on publication. Number of the companies I work with are fine that you own the data and you publish it. We just want an embargo of 30 days to get our internal messaging out before you release the results to the public. Some keep the data, and so you have to make peace with that yourself because you're doing it in that way. But we only very rarely will enter into those agreements. So those are the things you balance. NIH is a completely different animal. Obviously, that's all merit based. A lot of it that is convincing people, whereas industry always wants to test their products. For NIH, the balance there is often you have to convince NIH that this product, that this nutrient needs to be tested. So it's a really complicated soup of things. I don't think that I gave you a straight answer, but that's what I've learned.
No, that was quite helpful. I see there's somebody that Catherine has her hand up, her virtual hand up, but I don't know if there's people in the room or who's next. So Catherine, why don't you go and then I'll get out of the way.
Well, my question kind of piggybacks off yours, and I'm trying to decide if it's different enough. I was just, John, that was really fun to kind of see a whole career laid out for somebody who's at the beginning, hoping to see the end someday. And I'm personally very interested in measure development, and then, pivoting that into working with people for clinical trials. And I'm kind of wondering if there are other things in addition to what you said that, if you had to go back to the beginning, what are things that you sort of wish you would have known, or, wow, if we had known that this was going to go here, like, here's how we would have developed this measure differently, or I don't know if you have any sort of looking forward advice.
We stumbled around a lot early on. So there was, what I've done differently.
It's okay if there's nothing.
What I would have done differently is, I guess earlier in my career, I already kept asking why am I doing this? Why am I doing this? Because there were a lot of sort of very basic studies that I ran, and that that were sort of parametric things that really weren't that impactful. But I think really from the start of my career, if I had changed anything, it would be to focus on impact. And not so much on the quantitative aspects of your productivity, and more on like, does this really matter? And I gotta admit, being part of LSI and the Bureau of Child Research across my career really changed that for me. I was a very basic scientist, and not particularly interested in the application, and working around people like everyone in this room and everyone on this call really changed my outlook about that, and I owe that to the institution. I don't know, some strategies that I used early in my career is don't do anything unless it pays off. Don't go down the rabbit hole unless it was really meaningful to you in the long run. And if you do go down a rabbit hole, know when to stop, cut your losses, and then get whatever you can out of it. But I don't know if that helps.
That's great. Thanks.
Yeah.
We do a lot of assessments with traumatic brain injury, et cetera. See a lot of kids with ADHD, kind of following that very closely, not closely, having history. Have you done anything where you've taken a look at these kids when you knew what happened to them, supplemented versus unsupplemented?
Yeah.
Took 300, 400, 500, whatever, some number of these kids, went back and looked at them, and had someone do an assessment of whether or not they meet criteria or not for ADHD, for example, have learning disabilities.
Okay, so I don't know if you all heard Bob's question. It's about ADHD and kids with traumatic brain injury, and it's, can we predict that from infancy, but also do the supplements work for that
And look back.
and look backwards? Looking backwards, we did in the samples that we work with, we typically we end up with 1 or 2 or 3 kids with clinical problems later on. And so what we have been able to do is look backward and look at giftedness. So it's the other end. And that's an interesting study. I did a chapter with Francis on this. And what we found there was that that a kid who ends up in the gifted end of range, that range, they ascend to giftedness and they never drop back down. It's really an interesting piece. We don't see enough kids with disorders. We might end up with 1 or 2 or 3. And if you got 300, 500 kids, you must have a lot of money, Bob. Because we don't have that. What we have looked at, for example, are kids who are frankly disordered, bring them in and they have a genetic syndrome or something like that. And here's one thing. The one thing I've learned from that is all the things that we think are connected and are part of a unitary cognitive construct is wrong. A lot of what we see in terms of what we think all these things, like we see all these things in typically developing kids, and they all look like these things are unitary constructs, and they develop, and it's not. They all come together, and that's part of the typical development, neurotypical development, is that they're coordinated, they're organized. What we see in kids with Down syndrome is that they're all over the place. There's no organized or coordinated kind of attention that we see in Down syndrome infants. Getting to the supplements, there are actually lots of people ask me, oh, well, can you use DHA for autism? And the answer there is no. There's no good evidence suggesting there, but there is pretty good evidence, and it jives with the data we have here, suggesting that DHA positively affects attention. ADHD, there are 3 or 4 clinical trials out there showing pretty good effects on DHA on ADHD. And nutrients, I think, might be an important way to think about that. For example, even in cognitive aging, heavy doses of B12 will arrest the deterioration in mild cognitive impairment. So if you get people who are starting to show mild cognitive impairment, and you hit them with a massive dose of B12 on a chronic basis, that arrests everything. arrests the brain deterioration, it arrests those things. So, I don't know that it's as effective as a long-standing exercise, but it's pretty interesting there. So I do think there's a role for that there. I don't know if I answered your question.
Can I
Would be
ask-
oh,
Carrie has a question.
Sorry. I know
I'm
it's-
not cutting off Carrie. You go, Carrie.
Not needed.
Oh, that's nice. Yeah, I was gonna say, I know it's almost time, but John, you mentioned science communication and kind of this idea of getting to pediatricians as well as early on. I'm assuming that will, formula producers. There's also the element of, families and
Right.
others. So I'm just wondering if you could briefly maybe just hit on like your top recommendation around kind of that communication and science
So,
communication.
yeah, so I was in Switzerland 2 weeks ago. And this is a coordinated effort by Nestle. Nestle has a formula that they're getting ready. If they're listening, I'm in trouble because it's not, so it's supposed to be a secret. Nestle has a formula that they think is going to affect myelin. And preliminary results suggest that in fact it does. If you feed it early, it does show increased white matter. And it's been done by people I trust. And they expect to see the behavioral results in terms of executive function, improvements in executive function. So what the company is doing is it's getting out ahead of itself and developing educational materials to tell parents and healthcare practitioners what executive function is, so that when they release this and they say it affects executive function, that everyone will go, oh, I know that. So it's a coordinated effort. But in terms of science communication, I think this interface is what my job is dean too. I think everybody you don't have to do applied research. You don't have to be doing translational research. But what you have to do is you have to be able to say how what you do matters. Bottom line, you have to be able, in this century, we need public support, we need support of our legislatures, even when they're crazy. We need the support of the region, we need the support of our local officials. We have to be able to tell people why we are doing what we're doing and what they will get out of it. We have to, for example, we took papers here. I didn't show you the effects that we showed. We were able to reduce preterm birth. And so one of the things we did, and that was with another preterm, with a prenatal supplement of DHA1. That's one of the other things it does. And what we did is we published 2 papers on where we economized that effect and showed that this effect will save the US healthcare industry $7 billion over time. And that's just in the perinatal period. And we have to make those cases to society because we have to, and that's just something we have to do. How do we do it? We get rid of jargon, we explain things clearly, but we don't dumb them down. We try to get through it with a scientifically literate message that's digestible and intelligible. So there you go.
Easier said than done, but
But it's something we have to do. I would say.
absolutely. And you said it well. Well, thank you again, John, for wonderful talk. Appreciate everybody in the audience being here. And we'll be talking again in a couple weeks about the new Research Rising initiative that John, Carrie, and I have been leading. And so we invite you to join us again in 2 weeks. But thanks again, John. Thanks, everyone. Take care.